Girl, won't you stay?
Hello, Board of Directors. I am Dr. Ty, and this is my partner.
Marissa. Lim.
For our research, we're here to tell you about the differential roles of calcitonin family peptides in the dendrite formation and spinogenesis of the cerebral cortex in vitro. So basically, a little bit of background information is we started looking at neuropeptides for this, and the neuropeptide is a multifunctional chain of amino acids that act as a neurotransmitter. We've already found that some neurotransmitters have neurotrophic tendencies during brain development, and that is, neurotrophic is the development of neurons. And we've already looked into some, like monoamines, glutamate, and GABA, just some of the classical ones, and we found information on those in dendritic formation. And now we want to look at neuropeptides. So we examined calcitonin gene-related peptides, CGRP, and other calcitonin family peptides in vitro.
Okay, so a little bit about CGRP. It's a 37 amino acid peptide and is originally identified as an alternate splicing product of the CT gene, which is located on chromosome 11. It's produced in both the peripheral and the central neurons. And it has a quality of vasodilation, which means that it is responsible for the widening of blood vessels, for relaxation of smooth muscle cells. Its function is in the transmission of pain, so
it reacts with nociceptors.
Right, yeah, nociceptors. The messenger RNA and the protein of CGRP appear early in the brain during development stages. So the expression level increases briefly during postnatal days. So basically these expression patterns suggest roles in brain development, but only a few studies dealing with neural development have been actually done, so that's why we're doing this. So it promotes the differentiation of dopaminergic.
Dopaminergic.
Sorry, yeah, it was all her, so.
Of the olfactory bulbs and midbrain. So basically, recent studies have shown that the CPRG promote dendritic formations in the cerebral Purkinje fibers, which is one of the largest neurons in the human brain, and they're very well known for their very vast amounts of large number of dendritic spines. So if the CGRP is affecting the dendritic spines and Purkinje fibers, then it's possible that it is in other cells. So it's also a heterodimer, which is a protein composed of two polypeptide chains differing in composition in the order, number, and or kind of their amino acid residues, and it reacts with RAMP1. Some of the other ones we studied were adrenomedulin, which is AM, and it's a 52 amino acid chain, and it may function as a hormone in circulation control. So it's also another vasodilator, and it's one of the most potent in the body, vasodilation peptide in the body, and it's also a heterodimer, but instead it reacts with RAMP2 and 3. One of the other ones we studied are amylin, which is AMY. It's a 37 amino acid, which is the same as the CGRP, but it's just a different sequence, and it's co-secreted with insulin, and it plays a role in glycemic regulation by slowing gastric emptying and promoting satiety. But this one reacts with RAMP1, 2, and 3. The last thing we studied was calcitonin. It's got 32 amino acids, and it participates in calcium homeostasis by inhibiting calcium absorption in intestines, and it inhibits osteoclasts, and it stimulates osteoblasts, and it just overall reduces blood calcium levels, and it's a monomer instead of the other ones being heterodimers. And the reason we chose all of these is because all of the receptors are very closely related. They're 7 transmembrane G protein coupled receptors, and they all have ACLR, which is a calcitonin-like receptor, even though they do react with other ones like the RAMP1, 2, and 3, but they're all very similar. So the methods we used for this is we took the rat embryos at embryonic day 16 and 18, and we removed them from pregnant rats, and we quickly decapitated them with the cerebral cortex, and then took out the cerebral cortex. We did this with anesthesia for the rats, and then we removed the meninges and the cerebral cortex, and we completely incubated it in 0.05% trypsin EDTA for 5 minutes at 37 degrees Celsius. We took the day 18 embryo cortical neurons and we cultured them, in a medium and then was replaced by a serum-free neurobasal medium. And then 8 hours after doing that, we took each neuropeptide and we added it to the culture medium at the concentration of 10, 100, and 1,000 nanomoles. And then we removed proliferating glial cells and neuronal progenitors a day after plating. And we did this because we wanted to make sure that the only thing that was regenerating dendritic formation was the neuropeptides that we were putting in, and not anything else like astrocytes or any other kind of type of glial cells that might help dendritic formation. So to analyze dendritic protrusions, we took the 16-day embryos and we cultured it in a serum-containing medium for 24 hours and then cultured them in a serum-free neurobasal medium. And then at day 14 in vitro, the peptides were added for 24 hours. And then for each dendritic analyzation, we used immunostaining with the appropriate antibodies for each one.
Okay, so for our results, the only difference between the 2 and 3 days when CGRP is compared to the control is the increase of the branching index after 3 days. The peptides AM decreased the average dendritic length and did not affect the primary dendrites or the branching index. AMY decreased the total dendritic length and did not affect the primary dendrites or the branching index. CT had no significant effect on any parameters of the dendrite formation, and the treatment of AMY and AM inhibit the dendritic elongation of non-GABA neurons without effect on the formation of primary dendrites nor dendritic branchings.
Possibly help diseases like Alzheimer's, Huntington's, and Parkinson's, which are all based off the basal ganglion. And, Alzheimer's has been linked to the loss of dendritic spines, Huntington's to the link of abnormal dendritic spines, and then Parkinson's to loss of dendritic length and dendritic spines. So if we study this and we understand how maybe these neuropeptides affect dendritic formation in the early stages, maybe we can see, we can catch possible abnormalities in dendritic formation in the early stages that might lead to these kind of diseases later in life and hopefully stop them. Okay, Board of Directors, so if you give us this grant, a couple things that we could do different for the next time would be to examine the results of why the CGRP promotes the dendritic initiation and not just that it does promote it, but why specifically and how. And then we can combine samples of the neuropeptides instead of having them all separated, because in biological systems, the neuropeptides are all together and
not isolated.
They're not isolated by themselves. They work together, they interact together, and everything like that. And even further, even though there's a lot of ethical issues, further we could go would be to work on human neurocortical cortex dendrites.